Hello. Thank you for the opportunity to speak today. My name is Elaine Johan, and I'm the Director for Health Informatics within the Food and Drug Administration's Office of Data Analytics and Research. Omar Serang, the DNAnexus Chief Cloud Officer, and I will share with you how FDA is leveraging the precisionFDA multi-omics environment to inform regulatory science. As clinical applications for genomics continue to increase, the FDA is collaborating across government, industry, and academia to inform regulatory standards for multi-omics. In 2015, we were faced with the challenge of evaluating bioinformatic pipelines for the analysis and interpretation of next-generation sequencing instrument output. To respond to this challenge and engage internal and external experts, FDA launched precisionFDA as a collaborative genomics environment. In 2022, we are now utilizing precisionFDA to integrate other omics applications, including proteomics, viral and bacterial genomics, metagenomics, and imaging. This enables FDA to keep pace with the rapidly evolving state of multi-omics and data science. Today, Omar and I will be presenting precisionFDA and how it is built on a FedRAMP authorized DNAnexus platform as a service and AWS infrastructure to deliver an exceptional user experience for collaborative science. In particular, we will be discussing the Truth Challenge version 2, calling variants from short and long reads and difficult to map regions of the human genome. This competition was developed in partnership with the NIST Genome in a Bottle team. Through this example, we will show how real-world problems and regulatory standards are informed through precisionFDA crowdsourced competitions. precisionFDA is a secure, collaborative, cloud-based, high-performance computing environment where large data sets can be hosted, managed, and analyzed in a secure environment. It connects experts, citizen science, and scholars from all around the world. precisionFDA has over 6,000 users with access to over 100 terabytes of data and provides 60 uniquely publicly available apps, largely focused on genomics, but expanding into other omics and imaging. The precisionFDA user community embraces a wide range of stakeholders, including pharmaceutical, biotechnology and device companies, healthcare providers, academic institutes, research consortia, and patient advocacy groups, and of course, government agencies like the FDA. To date, the platform has hosted a total of 32 crowdsourced challenges and been involved with or referenced in 85 applications. We have also created 21 training and tutorial videos. In addition, the platform has achieved FedRAMP moderate status, and the portal has achieved FISMA moderate status in order to allow us to have secure, non-public collaborations such as those between FDA and regulated industry. precisionFDA delivers a fit-for-purpose user experience, providing user-friendly and intuitive access to manage cloud resources for a broad range of user roles, including academic, government, and industry researchers, FDA reviewers and pharma sponsors, expert programmers and data scientists, and even non-programmers. The precisionFDA web-based user experience is built on the DNAnexus platform as a service, served via the DNAnexus API. This interface layers critical components of a regulatory-grade platform on top of the underlying AWS infrastructure as a service, and includes data immutability and chain of provenance tracking, application versioning and enforced retention, FedRAMP authorized, regulatory-grade security and privacy controls for protection of PHI and PII. AWS provides the underlying FedRAMP authorized cloud services that are enhanced through the combined DNAnexus and precisionFDA stacks to provide the ultimate precisionFDA user experience, including Elastic Compute Cloud, Simple Storage Service, and Relational Database Service. But precisionFDA is not just a multi-omics platform and portal. It is a comprehensive service based upon a crowdsourcing model with users collaborating from 54 countries around the world, delivering capabilities that are constantly evolving to meet user needs and enhance collaboration. The wheel graphic on this slide represents some of the many capabilities that are offered, including the ability to work securely in a private workspace in the cloud, and also to collaborate in shared spaces, to build and import applications and workflows, benchmark them, execute analysis pipelines, and share the results in a reproducible manner. Publish applications and reference data sets for access by the general precisionFDA community, or even a controlled set of individual precisionFDA users. To participate in competitions and partner with precisionFDA to conduct competitions, engage in community discussions, and search substances and product relationships in the global substance registration system, which is integrated into precisionFDA. There are currently over 60 unique public precisionFDA applications covering a range of informatics domains, such as NGS, informatics, germline, somatic, microbial, genomics, transcriptomics, proteomics, metagenomics, HIV drug resistance, cheminformatics, and imaging. precisionFDA provides a framework that allows us to conduct competitions in a public-facing, high-performance computing environment. This scientific competition framework enables the FDA and partners to independently develop and present competition assets and applications, support challenge lifecycle workflows, and the submission and grading of competition responses from participants. Our competitions follow a lifecycle process beginning with inception and ending in post-competition activities, such as the development of a manuscript in a scientific journal. These competitions focus experts around the world on common problems in areas of evolving science, such as genomics, proteomics, artificial intelligence, etc. Often the post-challenge work to highlight the scientific discoveries and/or implement new tools in a real-world environment can continue long after the challenge. Through these scientific competitions, the FDA encourages innovations that advance best practices and inform regulatory science, enabling partners and participants to contribute to the bioinformatics, data science, and references that are foundational to the incorporation of multi-omics and real-world data into precision medicine. Over to you, Omar.
Thank you, Elaine. The FDA has actually conducted 2 truth challenges in collaboration with the National Institutes for Standards and Technology, NIST, one in 2016 and another one in 2021. Both of these challenges utilize previously unreleased data sets developed by the NIST Genome in a Bottle Consortium. The truth challenges leverage events in the scientific discovery and publication process to coordinate timelines of the scientific challenge to align with the release of information in the scientific process. And when presenting a challenge like this, the designers are really striving to provide sufficient information to the challenge participants to enable contribution, but to guard against overfitting of solutions to a particular data set. They also need to implement an automated grading objective. You really want an application that can automatically grade. You don't want a bunch of scientists sitting in a room subjectively grading participant submissions. It takes a unique confluence of events to have all the pieces of a truth challenge. You need multiple data sets that are similar enough to be able to enable them to be addressed with the same application. You need an unambiguous mechanism to compare submissions against the truth data. This fortunately has occurred not once, but twice on precisionFDA, with the variant calling truth challenge V1 back in 2016, focused on short read NGS variant calling and more simple types of variants, and the V2, which was presented in 2020, expanding into the hard to read regions of the genome, using both short and long read sequencing technologies. Let's take a look at the truth challenge paradigm in action. The focus of the variant calling truth challenge V2 was to advance our understanding of small variant calling accuracy by assessing variant calling pipeline performance on a not yet released, well-characterized reference data set, again developed by the NIST Genome in a Bottle Consortium. with a focus on difficult-to-map regions, segmental duplications, and the major histocompatibility complex. A novel aspect of this challenge is that participants could use whole genome sequencing data produced by one or more of Illumina, PacBio Hi-Fi, and Oxford Nanopore, the latter two being long-read technologies. The timeline started in May 1st, 2020, when 9 FASTQs were made available to the participants. These are essentially the unblinded set, and then the participants processed these FASTQs with their pipelines, and they ran a comparison against the NIST genome in a bottle processing. So the NIST folks, they've gone and characterized all these variant calls deeply with orthogonal techniques to come up with what they think is a reference standard. And so what they're asking for now is for the participants to see how their pipelines compare with the reference standard that the NIST folks have worked so hard to develop. Then the good part happens when the next set of samples are released to the public, and now the participants have their pipelines compared to a data set that they hadn't yet seen, again, to avoid against overfitting. And then the awards for concordance are made based on the comparison of the submitted calls from participants with what actually was called off of the HG003 and 4, the newly released sets. There are about 101 submissions for this challenge from pharmaceutical, instrument manufacturers, bioinformatics sector, and healthcare providers and research institutes. It was a very impactful challenge. So what did that lead to? We had a few significant outcomes. Several of the challenge submissions really outperformed the 2016 Truth Challenge. We see a significant improvement in short read-based variant calling. The variant calling pipeline performance was particularly good amongst the machine learning-based approaches that combined multiple sequencing technologies, like looking at both Illumina, PacBio, and Oxford Nanopore results. The machine learning-based methods also had larger performance differences between the originally released and the blinded set, which again indicates possible overtuning. Again, why you do a truth challenge, withholding a piece of data so that people don't actually see all the answers to the test. Several graph-based methods performed well, possibly by improving read mapping and alignment. These truth challenges have been well represented in scientific journals, including Nature Biotechnology, and most recently for Truth Challenge V2 in Cell Genomics. In fact, these challenges have been represented in more than 15 manuscripts. Scientific advances, publication, big deal for anybody working in this field. The Genome in a Bottle reference set had additional confirmation on their reference calls. This brought insight into important genomic mutations previously inaccessible. Commercial advances, objective demonstration of capabilities. There's no substitute for demonstrating your accuracy objectively with a government organization, a reference organization, no less. Publication and bragging rights is very important. Many of the people that demonstrated their superiority of their pipelines put that right on their websites. And then, of course, access to reference sequencing materials for bioinformatics application development. If you have these sequencing materials, these bioinformatics biological samples from NIST, you can put them through an entire pipeline and compare your results to the NIST reference standards. Last but not least, for the FDA, there are regulatory advantages from this. They advance and propagate the state of science for variant calling. We define levels of performance for regulatory acceptance, and there is community engagement for contributions that go far beyond FDA-only capabilities. So delivering a service like precisionFDA requires a cloud-based solution for a whole variety of reasons. AWS provides the highly reliable FedRAMP authorized cloud services that DNAnexus has built its platform for over a decade. AWS addresses all the infrastructure level provisioning, operations, billing, and compliance, and it frees DNAnexus up to focus on developing and delivering the high-value DNAnexus platform services that are the foundation of precisionFDA. The DNAnexus platform and scientific and security expertise spans key dimensions of life sciences, including diagnostic testing, pharmaceutical and biopharma research, trusted research environments like the UK Biobank, the City of Hope Cancer Care Network, and the St. Jude Pediatric Cancer Cloud, and of course, precisionFDA. DNAnexus' ability to present focused bioinformatics data and analysis services enables customers like FDA to develop rich solutions with authorized security and privacy protections in a very agile manner. DNAnexus is authorized under privacy, clinical, and drug development compliance regimes, including GDPR, FedRAMP, GXP, 21 CFR Part 11 for clinical trials, CAP and CLIA for diagnostic labs, and ISO 27001, which was sort of our foundation for security and compliance a decade ago. The FDA selected DNAnexus and AWS to stay ahead of the big challenges and rapid advancements in science, while maintaining the stringent controls required for a regulatory-grade service. DNAnexus and AWS have been partners for over a decade, delivering rapid, reliable, cost-effective realization of solutions spanning diagnostics, high-throughput, high-reliability analysis pipeline execution, real-world data creation and publication, cancer care networks, clinical decision support, and biobank research environments. We are not just a service provider to the FDA, we are a trusted development and execution partner, delivering an evolving, collaborative science platform to inform regulatory science. And back to you, Elaine.
Thanks, Omar. As Omar mentioned, the results of this very successful competition have been published in the May 2022 issue of Cell Genomics, where you can find a much more in-depth discussion of this challenge. Thank you for this opportunity to share the successes of precisionFDA. If you would like to participate in our competitions or learn more about past competitions, please go to our website at precision.fda.gov. If you would like to partner with us on the creation of a competition, please reach out to us at precisionFDA@fda.hhs.gov. Partnering with DNAnexus and AWS, our use of experiential learning has benefited the mission of FDA, developed the AI skills of our workforce, and provided real-world solutions that reach beyond the boundaries of FDA. Thank you so much for your time today.